Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Hi, everyone. Welcome to the Genetics Podcast. I'm excited to try something a little bit different and hopefully fun today, which is I'm going to do a solo episode where I'm diving into a topic that I've been spending a lot of time reading about, thinking about. It's in the news as well, so I can't help but think about it every couple of days as I see articles about companies that are exiting the space. And I'm talking about gene therapy, and in particular, AAV-based gene therapy. So if you're following the news, you've probably seen over the last 6 to 12 months news about companies pulling out of investing in gene therapy. For example, Vertex, Pfizer a while ago, Biogen very recently. But also there have been articles about growing safety issues, although this is with a growing number of patients being treated. So I'm going to talk a little bit today about what we know about the safety issues with gene therapy, where they're showing up, where they're not showing up. But for example, Sarepta Therapeutics was widely reported on 3 patients dying, 1 in clinical trials and 2 for an approved therapy. And so this is a topic that's on a lot of people's minds. And in particular, as times are a little bit more challenging right now commercially in biotech and pharma than maybe they were 3 or 4 years ago, we're starting to see a lot of prioritization. And this doesn't mean people are pulling out of genetic medicine and precision medicine in general. They could be refocusing on other delivery methods of gene therapy like nanoparticles or other next generation methods. They could be focusing on RNA or just focusing on biologics and small molecules, where there's lots of other areas that with a good understanding of the fundamental biology, you can make a lot of impact even on the rarest diseases. So we're going to talk a little bit about this today because I think it's important to understand where we are, but also some of the scientific bottlenecks that, if they're cracked, mean that people might come back to this. All right, so with that summary and overview, let's jump into it. I want to start with actually just defining gene therapy first. So there's a few different definitions of this, but one that I think is clearest and will help us with this conversation is basically any therapeutic that introduces, removes, or alters genetic materials within the cells at the genetic level is what I'm talking about today. So what importantly this takes out is things like RNA interference or other RNA-based methods that don't achieve a durable or permanent therapeutic effect. But what we are talking about is introducing a full copy or a partial copy of a gene using AAV or some other kind of viral vector. We also can be talking about gene editing, making a permanent change. But I'm really going to focus today on AAV gene therapy. But I just want to start with that starting point. And what we're focused on here is replacing a missing or defective gene, so something that is broken and not working. A lot of haploinsufficient diseases, for example, fall in this category. It could also include adding a new gene that has some kind of restorative or protective function. There's a little bit less of that, but it's absolutely a concept that we can think about and talk about today. And then also editing or correcting a gene in some way. So that's what we're going to talk about. And another maybe an analogy to think about here that could make this more helpful is you can think of this as gene transplant. It's a little bit of an oversimplified analogy, but I think it's a really effective one. We're going in and bringing in a functional copy of a gene in order to restore all or some partial level of gene expression. That means we can either halt or reverse some kind of pathology. The reason I think gene transplant is also a useful metaphor is because you also have to think about immunosuppression, at least for a period of time, because of the immunogenic nature of viral vectors in particular. So, we have this paradigm to think about, and I'll try to carry this through the episode as we think about what the challenges are and what the opportunities are to mitigate those. So diving into AAV, where we're seeing, historically, we've seen a huge amount of investment and activity here, and it's delivered some very remarkable therapeutics. We're going to talk about that, but also it's attracted a lot of attention lately because of a number of high-profile companies pulling out of AAV after spending hundreds of millions or billions of dollars in research there. So what AAVs are, it stands for adeno-associated virus, and they're small viruses that basically can package up DNA and deliver those into all kinds of different cell types. This is both a feature but also a bug of this approach, because viruses are very good at getting into the genome, getting into cells. We can get them to lots of different parts of the body: cardiac tissue, heart muscle, skeletal muscle, liver, brain, eyes, almost every organ. You can think about how you get there with a viral vector, either through systemic administration or going directly into the organ. But that also means that you have to think about off-target effects of how they get into organ systems that you don't want them to go into, like the liver in particular, where a lot of the issues are caused. And so with this in mind, there's a lot of next generation AAV approaches that use evolution, either directed or machine learning based, to try to overcome these, to try to overcome these challenges. So to get them to be more tissue specific, to get them to be less immunogenic. And so we'll go on to that in a little bit of time, but we're going to focus on AAVs. They're one of the workhorses of gene therapy because they're non-integrating. So they don't actually integrate into the genome, which is a good thing because they are extremely durable, especially in cell types that don't divide very frequently, but you don't have the as much of a risk of off-target effects compared to something like lentivirus or another vector that is integrating into the genome. So I'll dive into an example here that is using AAV and has been a major success. So that's SMA, spinal muscular atrophy. This is a neuromuscular disease that's caused generally by loss of function in the SMN1 gene. That stands for survival motor neuron 1. So SMN1 provides the instructions for making the SMN protein, and this is an essential building block for the survival of motor neurons. So these nerve cells basically degenerate, lead to progressive muscle weakness, and have severe adverse effects, including death in infancy and terrible prognosis and unmet need in this population. What is important that I want to cover about the biology here before we go on is we also have a backup gene, effectively. It's called SMN2, but it only produces very small amounts of functional SMN protein because there's a splicing issue. And this is important because the gene therapy goes in and simply replaces SMN1 and provides a functional copy of the gene. But there are other approaches actually that relate to fixing this splicing error in SMN2 that I'll talk about a little bit later. So Zolgensma is the AAV-based gene therapy. It's developed by Novartis, and it's a one-time treatment that goes after this missing SMN1 gene. And this uses AAV9, which is one of the many AAV options that we can think about, but it's really the most common. We can talk about some of these other options that are used, but what this does is brings a fully functional copy of the SMN1 gene, and it has a promoter that's specific or not totally specific, but it's really tuned to expression in motor neurons. So AAV9 can cross the blood-brain barrier. It gets to spinal motor neurons with just one single IV infusion. It can go to other parts of the body, and it does go to the liver in particular, and there are liver toxicity issues here that have been well documented, but it is under a promoter that's really tuned to motor neurons. So you get this functional restoration of the SMN1 gene in the motor neurons, and the data is really remarkable here. Especially the earlier patients are treated, the better outcomes are. So this has been a major success, and basically the way that it works is, as I described before, the restored protein prevents the degeneration of the motor neurons, and so as you could probably imagine, the earlier you can go with delivery here, the better the outcomes are likely to be. And I'll just stress here that it doesn't actually replace or edit the existing DNA. It just adds in a new functional copy of SMN1. I want to just talk about the other options that are available here, because this is going to be important later as we talk about why people are moving out of AAV and what it might mean for the future of the field. There are two other approaches that actually go after, that I'm aware of, at least two other that are on the market, that go after this same issue, but with a different approach that's focused on the splicing error in SMN2 that I talked about. So Roche has a small molecule that basically promotes inclusion of exon 7, which is the one that is spliced out and results in not very much functional protein being produced. By promoting inclusion of that exon, you can actually effectively rescue the phenotype by getting more SMN protein produced by SMN2. And Biogen's drug Spinraza is a similar mechanism, but it's using an oligonucleotide approach to promote the exon inclusion for SMN2. So these are three therapies that are on the market for this disease that a decade ago had very little options. So whether you're on whether if you or your family is experiencing this disease, and whether you're on small molecule, oligo, or gene therapy, I think it's pretty clear that understanding of the fundamental biology, but also some of these next generation therapies are meeting a huge unmet need here. So to go on to another example, which is one I mentioned earlier at the top of the show, is in muscular dystrophy. So I'm going to talk about Sarepta, which it's been in the news a lot lately due to these safety issues that I mentioned. So just as a disclaimer here, I don't have any information on this that's inside. This is just based on what I'm reading in the public domain. As we talked about with SMA, there's really three things to think about with these therapies. There's the vector, which is what is the delivery vehicle to actually carry the genetic material into the cells. There's the promoter, so it's the regulatory element that drives the expression, ideally in as specific of a cell type as possible to have the desired therapeutic effect. And then there's the transgene, which is basically the payload. What DNA sequence are we trying to get into the cell type that we're going after? So for Sarepta, like the Zolgensma case that we just talked through, they're also using AAV, but they're using a specifically designed variant of AAV called AAV-RH74, which has a very favorable muscle targeting properties. So again, like the AAV9 that we discussed before with the motor neuron specific reporter, this has a real specificity to muscle, but it does not prevent it from going into the liver and into other places. It's very hard, it turns out, to get it to go very, very specifically to a single muscle cell, for example. So they've done a ton of work to tweak these promoters, look at different ways to try to ensure that expression primarily or only occurs in certain skeletal muscle or cardiac muscle cells. So there's a lot of work that goes into this. I'm not going to go in depth into promoter design here, but I'll talk a little bit later about some of the areas of development in vector design more generally. Something that is a little bit different about this program is the dystrophin gene for muscular dystrophy is an enormous gene. So we are actually not able to fit the full gene into the AAV vector. So they effectively introduce a truncated version that still produces a functional protein. So it's just an important detail here. But this is also one of the drawbacks of AAV is the payload size is limited. You can't get huge genes like dystrophin or titin that's very relevant in cardiomyopathy into an AAV vector very easily. So this causes people also, besides the issues that I'll talk about around safety and scalability and commercial viability, this is another factor that may sway someone to think about focusing on AAV or focusing elsewhere. So diving into Sarepta's gene therapy for muscular dystrophy, the way that this one works is mutations in the dystrophin gene prevent the muscle cells from making the right amount of the dystrophin protein. It causes issues with muscle integrity and causes the progressive weakening that we see over time. Their therapy is intended to deliver, as I mentioned before, a version of the dystrophin gene that isn't the exact copy of the functional gene, but is a miniaturized version that creates enough functional protein to help restore those muscle cells. So once it gets inside the cell, the transgene is expressed, and the cell starts to produce this dystrophin protein and restores muscle function. That, like the Zolgensma example before, it's given as a one-time infusion, so one dose. And importantly for this one, it was initially conditionally approved within certain Duchenne muscular dystrophy populations. And most importantly for our discussion today, it was initially approved for ambulatory patients. And this has been under close review. The way that these conditional approvals work is they're allowed to go into the market, but there are additional requirements for data collection over time. And the company works very closely with the FDA to monitor not just long-term safety, but also efficacy. So getting into the recent challenges that I'll be paraphrasing a lot of the news that you may have read, but I'll try to go into detail if you haven't read any of this news yet. There have been some challenges with this, namely around liver toxicity and acute liver failure. So across almost all gene therapies, there are issues with liver toxicity. So this is not by any means unique to the Sarepta program. the fact that this is a virus and the fact that it makes its way through and it makes its way through to the liver results in a lot of load on the liver that can show up in patients as major liver toxicity side effects. So obviously we have to think about a few factors here. The first is these are often very sick patients to begin with, and so the additional load on the liver can be problematic in combination with all of the other issues that the patient is having. It can also make it difficult to pinpoint exactly what the cause is. As we mentioned before, there is also immunosuppression going on. There are a lot of factors that go into this, but one thing that is not disputed is that AAV puts a lot of extra load on patients' liver. And this is because it's basically the filtering organ, right? It's taking everything that passes through the body. And so any high doses of virus or off-target uptake in the liver cells can contribute to this liver injury. So around middle of this year, the FDA placed a clinical hold on one of their clinical trials, and also there was that for a small period of time, the treatment was put on hold and was not commercially available. This was pretty quickly turned around, and it seems like Sarepta has been working with the FDA to try to share more data around what's happening and what populations. So to dive into this a little bit directly around what we're seeing here, I'll just quote directly from a Fierce Pharma article because I thought it was pretty well stated and gives us some parameters on what the issues that we're working with here. So what they say in this article, and I'll link to it in the show notes, is that liver toxicity, a known side effect of ELEVIDYS because of immune responses to the drug's viral vector, has become an even more concerning issue after 2 deaths were reported in March and June, plus a 3rd death tied to an investigational muscular dystrophy drug using the same gene therapy technology. All 3 patients were not able to walk because of the stage of the disease. So this is not saying that the drug caused them to not be able to walk. This is treating non-ambulatory patients rather than ambulatory patients. So again, going back to quoting Fierce Pharma, for ELEVIDYS specifically, among the 1,043 patients treated with the gene therapy in clinical trials or the commercial setting worldwide, the overall rate of hospitalization due to acute liver injury was 5.8%, according to Sarepta's analysis for the data. After ELEVIDYS's initial FDA approval in 2023, 5.8% of 552 ambulatory patients and 8.4% of the 107 non-ambulatory patients were hospitalized due to acute liver injury, according to the company. So what we're hearing about here is in the ambulatory patients, 5.8% of those experience acute liver injury, according to Sarepta, and 8.4% of the non-ambulatory. So there's a greater effect in these patients that have more severe disease and are not able to walk at the time of treatment. And this gets to probably the point I was making earlier about the additional comorbidities and the load on the patient's body as they start to become more and more progressed. A little bit later in the article, it also says that the acute liver injury hospitalization rate caused by Sarepta is lower than the 9.8%, which was estimated by Citi analysts using the FDA adverse event reporting system. When Citi came up with this number by counting all hospital admissions that overlapped with acute liver injury, Sarepta's calculation appears to be restricted to patients who were admitted specifically for acute liver injury. So we have a range here, I think. Some independent analysis had put this up to 10% of patients. Sarepta's own numbers, 5.8%, 8.4%, depending on the way that you cut the data. So this is a significant fraction of patients, and a fraction of these patients who are hospitalized for acute liver injury, unfortunately, go on to die. And so this is at the center of the issues here. And I think this is obviously a huge challenge to overcome when you're designing these therapies and you want to make them both effective but also safe. But it adds to the complexity of running trials for patients. It adds to the complexity of running trials for sponsors. And so all of these start to produce some headwinds in AAV gene therapy development that I think would, for obvious reasons, cause companies to take a pause and think about some of these other options that they have, like RNA, for example, that may not have some of these issues, but also other delivery methods.
The Genetics Podcast is going live at ASHG 2025. Join us in Boston on Tuesday, October 15th from 12 to 1 p.m. in room 151A for a special recording with expert guests from academia and biotech on the future of genomics. Lunch will be provided and seating is first come, first serve. If you're attending ASHG, we hope to see you there.
Okay, so in addition to the safety issues that we've spent a lot of time talking about, there is another compounding factor that's related in many ways, but is also a distinct issue, which is the commercial viability of these programs. So, and we need to talk about two things with this. First is the size of the patient population. So SMA, muscular dystrophy. We haven't talked about Luxturna, which is the very first gene therapy that was actually ever approved, which treats an inherited retinal dystrophy. These are all rare or ultra rare diseases, and so the market size of these is not unlimited in the way that GLP-1s for obesity, or or broader heart failure, or something like dementia would be. And so the challenge that we have here is you have these issues on the safety and scalability sides, but also when companies are looking at these therapy areas, unfortunately, they can come to some pretty sobering conclusions about the size of the market. Maybe as an illustrative example here, so the SMA that we've been spending a lot of time talking about is by almost anybody's measure a very commercially successful drug. It's done several billion dollars in revenue. More than 3,000 patients, I think, have been treated by it historically. But it is actually, from my understanding, probably the third most successful in the category. The two that I mentioned earlier, the small molecule and the oligonucleotide-based approach that go based on exon skipping and SMN2, have been more commercially successful historically. And so even in these major success cases, we see that there are potentially viable alternatives that can go after these opportunities. This is also the case for muscular dystrophy, for example. There are exon skipping therapies and other approaches besides gene therapy that have potential here. So for some of these diseases, there is, especially the ones where there's a large market opportunity, there may be underpinning biology that means you have multiple ways to go after the disease besides gene therapy. And this is a good thing ultimately for patients. We want options. We want, especially in the case of gene therapy, I'll talk about this a little bit more later, but it's a one-time treatment. That means you may not be able to retreat patients in the future with another AAV-based gene therapy. And so having a gene editing based approach or an RNA based approach or something that you can stack on top is potentially attractive as well. So, but these are small populations for the most part today. There are gene therapy programs in development for much more common diseases. So genetic subtypes of Parkinson's disease, age-related macular degeneration, type 1 diabetes. These are early clinical, or in the case of type 1 diabetes, I think it's actually preclinical. But there is a lot of work going on here to try to open this up to much bigger populations. But there is a second challenge here, which is the cost of goods here, the cost of developing the vectors. You need to grow vats and vats of virus, fill them with payload, and it's still very expensive on a per dose basis to develop these therapies. Now, this is coming down a lot, and I suspect that as more and more common diseases move into the pipeline, you're going to get continued market pressure to develop more and more scalable methods of developing these viral vectors. So I think the data shows that these continue to drop in cost over time, but they are still far more expensive in most cases than small molecules or biologics or other things that we've had decades to optimize manufacturing processes. So I don't see this to me, feels like an engineering problem. And there's scientific challenges in here as well. How do you grow? How do you make more viruses contain enough payload we have this empty capsid problem, basically, that I'll talk about in a little bit more detail now, actually. But how do you fill viruses? How do you fill enough viruses with payload that you're getting as much of the gene as possible into the right cell type, and you're not delivering empty viruses that basically exacerbate this toxicity or immunogenicity issue without actually getting the benefit of delivering the gene? So there's a lot of scientific and engineering work that can be done here. But what this contributes to is there's added cost on the clinical trial side and safety management and making sure that patients are protected. There's also added cost on the cost of goods side to actually develop these therapies. And if you couple that with a small market, then it makes for a very challenging spreadsheet for someone to put together and invest the billions of dollars that it takes to get a therapy to market today. But like I said, there is hope here because the science is continuing to march on. There's a lot of work that's being done in reducing the cost of goods and also in innovating on vector design as well as other types of delivery methods. Okay, so I'm just going to take a moment here to summarize where I think we are in terms of the benefits as well as the issues. So there are some very clear benefits. We have a technology that can address unmet need and provide, incredible treatments, or in some case, a functional cure for diseases that never had this possibility before. So this is impossible to understate how exciting this is when it works. There's also a major benefit of one-time treatment. And depending on how much growth there is in the tissue, how often the cells divide, this can be very durable and long lasting. And so if you just think about these two benefits, there's a lot of diseases where this has potential to be extremely impactful, and either has been already or will be in the future. But to summarize the issues, we see fatal adverse events and liver toxicity in particular in many gene therapy programs. This is the more research we do here, the more we understand the underlying drivers to this. But this is a huge factor when patients think about these therapies, and it's also a huge factor to consider in clinical development. There is also immune response to the vector. So this is what underpins the liver toxicity, but it also can result in other kinds of immune responses that are involving the factor C complex and other immune responses that are separate from. So if I then just summarize the challenges and where we are today, we have the immunogenicity problem, the fatal adverse events, and liver toxicity. This is happening in almost every program to a greater or lesser degree because the unmet need is so high here, and because so many patients that are in these programs are fragile to begin with. This is a really complicated calculus of, if you have something like SMA, how much do you accept liver toxicity and fatal adverse events given the natural course of disease? As we move into more and more common and potentially less severe diseases, this risk tolerance changes, but it's fundamentally a hard problem to balance. And the more that these liver toxicity issues persist, the more challenging that is for drug developers to wrap their heads around. We also have durability and expression issues. So especially in cases where children, for example, are treated early, and if it's in something like muscle cells or some part of the body that's going to grow quite a bit, because these are not replicating and not integrating, they're not integrating into the genome, you will, just from a first principles approach, potentially lose some of the effect over time, because as cells proliferate, you're going to lose expression of the transgene on a relative basis. Now, in any particular disease, whether there's a dose response early in development, later in development, how much that matters is really going to depend. But as I mentioned before, we do have to think about this not in isolation. If you have a gene therapy that works really well and isn't perfectly durable, if there are other second or third line therapies that can come online to help redose with a different delivery mechanism, or use something like a small molecule or biologic or oligotherapy to carry on maybe later in life as the durability starts to wane, I think we can think about these as part of a system, not in isolation. And then the final challenge here is around manufacturing, cost, scale, purity. Going to more and more of these gene therapy conferences, it's amazing how much work actually needs to go into the manufacturing of the vector. There's a huge amount of technical complexity here. A considerable fraction of the viral particles are actually empty. There's no payload. And so these are effectively carry this additional immune load and viral toxicity, but without delivering the payload and having the potency. So there's a lot of work that's being done here. I'm going to dive into this in a moment, because this is one of the areas where I think continued research and continued work is actually going to unblock some of these other issues in a dramatic way. So diving into that, if we talk about delivery methods for a second, there's a lot of real world impact and potential here. But if we focus on the two issues of liver toxicity / immunogenicity, and also the high costs of manufacturing these therapies, then this leads us right to the delivery methods, because the underlying vector here is what's driving the costs and is what's driving the immunogenicity. So how does it work today? It's a huge area of research in terms of designing the viral vector capsids. So there is no one AAV. There's AAV9, there's AAV8, there's AAV5, and there's all kinds of evolved versions of these that biotech teams around the world are working on to make them more tissue specific and less immunogenic. So the way this works at a very high level is you start with a diverse pool of capsid variants. So You normally would start with one of the, AAV serotypes that you're interested in. So if it's getting into cardiac tissue, maybe it's AAV8. If it's getting into brain, AAV9. There's different choices here. And I'm by no means an expert in AAV serotypes. So please don't hold it against me if I get a few of the details wrong here. But what then happens is there's different methods to basically apply selection pressure and evolve these capsids. And this can be very directed in the sense that if we understand biologically a tissue-specific promoter, for example, that we believe will make this specific to the motor neurons or cardiac tissue or something like that, you can engineer that promoter in and potentially introduce mutations or evolve the capsids in some way, using selection pressure to select ones that are more tissue specific or less immunogenic, or obviously in combination, we want them both. The challenge is that it's not easy to do all these things at the same time. As you iterate through the evolutionary process, whether it's directed or something that's a little bit more machine learning or AI guided, you're trying to balance potency, specificity, manufacturability, and scalability, and these immunogenicity and toxicity factors. A lot of this can be measured in vitro, but not all this is necessarily knowable a priori as well before you go into patients. So there's a lot of moving pieces here. And just to summarize, this is a huge area of active research. You'll see if you follow this space like I do, or you start to follow it after you listen to this, you'll see a lot of announcements between biotech companies, pharma, around these engineered capsids, whether they're brain specific or cardiac specific or designed in some way to be better than the naive AAV9 or AAV8 that the first generation of therapies are based off of. So there's a lot of work going on in AAV in general. Okay, so there are also other delivery options that I want to spend some time talking about. I won't be able to go through everything out there, and I'm confident I'm going to miss something. And please let me know if there's something really cool and interesting like DNA origami or something like that I'm missing. But I'm going to try to go through the main workhorses. So lentivirus and retrovirus are RNA viruses that convert their genome into DNA and then integrate it into the host cell. So unlike AAV, these do integrate into the cell, and they're used very heavily in CAR T therapies, stem cell modification. They're very stable and can be expressed for a very long time because of this integrating nature. And that means as cells divide, they keep this, unlike the AAV, which is going to be lost or diluted as part of the cell division process. But as I mentioned earlier, integration carries risk. There could be disruption of the existing host genome, integration where you don't want it to be. And so generally, these are not used as wide. They can't be used for everything, basically, is what I'm trying to say. There is also options like adenovirus, which is different than AAV, adeno-associated virus. They can take very large payloads, so much bigger genes, but they're highly immunogenic. So these are actually what's used in the COVID vaccines, or many of the COVID vaccines, not all of them, obviously. J&J, AstraZeneca, they're using adenovirus to deliver their payload, and they're good for short-term expression. They're not to be really used at high dose, so they're really quite good for vaccines because they're immunogenic. They trigger that immune response, which is actually what you want in the case of a vaccine, and so adenovirus is used a lot in that case. If we think about alternatives in some of the areas we mentioned, muscle, brain, cardiac tissue, one of the areas under a lot of active research is lipid nanoparticles. So, and these are also used for mRNA vaccines, obviously. The big advantage, as I see it, about lipid nanoparticles is they're not a virus. So if you can get them to be accurately tissue targeted, if you can get them to have long-term expression, then you potentially avoid the immunogenicity problem because you're not delivering with the virus, you're delivering effectively with a lipid bubble that carries the mRNA, DNA, CRISPR components, whatever it might be. And so for today, these are used for relatively, short mRNA vaccines, for example, are a great example of this. And you can actually repeat dose with these, and you will probably need to because of the transient nature of them. But they probably, in the current form that they are not a like for like swap for AAV because they have that transient expression. And the tissue targeting is limited, mostly liver today, but there's a ton of people working on ways to get lipid nanoparticles into other organs or organ systems. There are a few other things to think about. I'm not going to go into detail, mainly because I don't know very much about them, but DNA and RNA electroporation, so basically electric pulses to to open up the cell membrane and get the payload inside. There are also these kind of interesting virus-like particle systems. So they're inspired in a way by viruses, but they're not actually viruses, and so they're basically fully synthetic vectors that are inspired by viruses that can mimic the machinery that viruses use to get into cells, but with more, greater control over safety and the immunogenicity issue. So just to summarize where we are today, I mentioned at the top of the show, we're seeing a lot of people announcing that they're pulling out of AAV gene therapy. And this is, as far as I can tell, for many of the reasons described above. There's the immunogenicity challenges, there's the scale and cost of good challenges, but something that is really important to be explicit about, that many of these companies are being explicit about in their announcements, is that they're not pulling out of gene therapy entirely. They are tactically retreating in a way from AAV as a delivery method. They may still be focusing on some of these other delivery methods. They may this may be a nice way of them saying we're getting out of gene therapy entirely because AAV is such a big portion of it, but we're going to keep doing some work and may come back in the future as things change. So, I'm keeping an eye on this because AAV for a long time has been the workhorse of this industry, and seeing so many people start to pull back from this is definitely notable. But at the same time, we're also seeing many people stay the course, double down on their investments, especially the ones that have been successful here historically. Okay, so where do we go from here as we wrap up? And where are we today, really? Where is this working? It seems like much of the progress has been made and probably will continue to be made in parts of the body that are immunoprivileged. So the brain and the eyes, for example, are two areas where there's been a lot of gene therapy success historically, and I think there will continue to be going forward, because these are parts of the body where the immune system is just not as active, and they're cordoned off from the rest of the body in a way that other organ systems aren't. And so if you can deliver with a vector that crosses the blood-brain barrier, or you can deliver directly through, directly to the brain, for example, then these immune issues can be less of a problem, because if you combine this with a promoter that's very specific to the tissue of interest, then you have the opportunity to effectively deliver this therapy a little bit more locally rather than systemically. So we spoke about CNS and spinal muscular atrophy, central nervous system, but also Luxturna, which we didn't cover too much, is an example of a really durable and effective treatment in the eye. But this did suffer from some of the commercial issues that I mentioned before. There was possibly an overestimation of the size of the market of people that could be treated here. So while it was absolutely a stunning scientific success, I don't think it's fair to say it was a major commercial success. So my prediction, if you'd like to hear my prediction, is that we're going to continue to see a lot of progress in these parts of the bodies that are immunoprivileged, because the downsides that I mentioned here are greatly mitigated. They don't disappear entirely, but they're significantly mitigated compared to other parts of the body. What I also think is we're going to continue to see a lot of work go into engineered capsids, and I believe in the power of science here, that if we can get better at solving this off-target immunogenicity problem and get better at delivering these payloads to the tissue and maximizing the amount of gene that's delivered and minimizing the amount of virus delivered, we're only going to get better at managing these side effects relative to the impact that these treatments can have. So this is maybe driven by breakthroughs, but probably more accurately just driven by really grinding on what we know and developing better vectors that take the failures of the last generation, learn from them, and integrate them into the next step. I also think that, and probably driven by the focus on the immunoprivileged parts of the body, if we can see these gene therapies start to get to scale in more common diseases, I'm thinking about Parkinson's, GBA Parkinson's, for example, cardiomyopathies, frontotemporal dementia, maybe Alzheimer's. I didn't mention this, but I saw an interesting preclinical presentation about delivering the protective Christchurch variant with gene therapy into Alzheimer's patients. That's probably a ways from the clinic, but what I'm getting at here is if we start to see more common diseases being treated by gene therapy, then this is going to be a natural market push to drive the cost of goods down. Manufacturers are going to figure out how to produce these at much greater scale and much greater efficiency. And this should help drive the cost down for rarer diseases of the eye and central nervous system. So that's my prediction around those parts of the market. And I think, obviously there's low likelihood that people retreating from AAV are spelling the end for this. I think it's probably related to the focus of their pipelines and what they're working on and how they're managing their resources right now. But I think it is also a signal that we need to think about how to solve these critical and potentially systemic issues that are getting in the way of really scaling gene therapy. So the final note that I just want to make here as I dig into more detail, what is fascinating as well is also, once you really understand the biology fundamentally of a disease, it may be that you have options that wouldn't seem so simple initially, but actually turn out to be fundamental and extremely impactful. So gene replacement for a haploinsufficient gene is always going to seem like the most straightforward way to treat something biologically. And if we can make that work, it absolutely makes sense. If you should have two copies of the gene, you only have one, you can introduce, enough through a viral vector or other delivery to compensate for that dosage, then it's very rational that would, especially if delivered early and pre-symptomatically, potentially, dramatically delay symptoms or potentially cure altogether. But the more we learn about biology, I think the SMA example is a great one here. We have a compensatory mechanism that we can go after with more tried and true approaches, small molecules, oligos. And so for the genetics community, I think something that is interesting to think about is, as with the monogenic disease genes that we discover, the more we can invest in understanding the compensatory mechanisms or the gene networks around them, there's probably a lot of opportunities for synthetic lethality or looking elsewhere in the pathway that the gene sits to discover a way to, if it's not safe or scalable or cost effective to go after directly with a gene therapy, how can we go after it indirectly with some of the tools that we have a little bit more, ability to scale cost effectively and safely today. So I hope this was interesting. If you liked this and would like to hear more, then let me know. There's other topics I could probably do solo episodes like this on. If you didn't like it, then that's fine as well. I won't hurt my feelings. Just tell me you like to hear from other people and we'll focus on that. Thank you, as always, for listening, and we'll see you next time. Thanks, as always, for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate it if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy-Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.